So now we're moving a little further on in the fasting state, and this is a particular state that hopefully we don't have to get to. But I want to point out that ketones being made in ketogenesis is a good thing. Now, medically, you know inherently that this is something you have to deal with people that have either poor feeding habits and suffering horribly from malnutrition, or somebody that is an uncontrolled diabetic, ketoacidosis. So ketones are clinically a bad thing. But the reason that they're a good thing is because they're designed to keep us alive long enough to find food and to reverse the metabolism that's changing. Now, when do ketones come about? Well, if this is our glucose production early on is maintaining our glucose level, and this, of course, is glycogenolysis plus gluconeogenesis. Now, these two metabolic activities are what keep us going under normal circumstances. And normal circumstances are primarily because we are hopefully not going into a very long-term fast or starvation state. People who fast for 24 hours, even 48 hours because of religious requirements, they're fine. But pretty much all the religions, they will also warn their people that if you're going to fast, you may want to take some juice. You may not want to have a complete fast. And the reason for that is because they know about ketoacidosis, going too far and too long. Now, what we're talking about here are days. So up until about the point of 2 days, 48 hours, our glucose levels are being maintained at this point primarily by gluconeogenesis. But at this point, our glucose production will drop. And it will remain steady until either we eat or we pass. Now, this particular drop in gluconeogenesis is primarily being compensated by two other molecules. And one is this, and this is called acetoacetate. And there's one that's even being made more. This is called beta-hydroxy butyrate. And these two are the ketones. Those are the two ketones. Now, the important thing about ketones, two major things. First of all, they are highly energy rich. And so these molecules can be synthesized. They're synthesized by the liver and only by the liver. So they can be used by all cells except the liver. The liver does not have the capacity to use these ketones for energy, but all cells can use them in order to generate ATP. That's the good part. The bad part are the last three letters, ATE. That indicates that these are acids. So that's the downfall of these molecules. So how does the liver make this? And what is this metabolic shift that occurs in order to generate ketones and drop the rate of gluconeogenesis? So we're in the liver mitochondria. And the source of ketones is going to be acetyl-CoA. Now, acetyl-CoA in a fasting starvation state in the liver is coming from what energy source supplying this in a fasting state in the mitochondria in the liver? Fat beta oxidation, yeah. So this is beta oxidation generating our acetyl-CoA. Now, what we're going to do with the acetyl-CoA is we've got two uses for this very, very high level, high concentration of acetyl-CoA. The first thing that we're going to do with this is to inhibit pyruvate dehydrogenase and activate pyruvate carboxylase. And of course, this is going to provide us with the resources we need for gluconeogenesis. Now, once the acetyl-CoA levels rise to a certain level, now we will have a sufficient amount of substrate for ketone synthesis. We're in the mitochondria of the liver. What we're going to do is we're going to take 3 acetyl-CoAs and we are going to make HMG-CoA. There's no purposeful activation of ketone synthesis. What activates ketone synthesis is simply the concentration of acetyl-CoA. As this concentration goes, now we can start filling in this pathway. HMG-CoA, this is the hydroxymethyl-gluteryl-CoA that we saw is for cholesterol synthesis. But, kind of trick question, this is in the mitochondria. If this was in the cytoplasm, then that would be used for cholesterol synthesis. Seeing this in the mitochondrial matrix, it's for ketone synthesis. Now, the next step of this reaction is different than cholesterol synthesis. So cholesterol synthesis is going to require the reductase enzyme to modify this to mevalonate. So the HMG-CoA reductase enzyme is not present in the mitochondrial matrix. It's only in the cytoplasm. So we are not going to perform that reaction here. What we have is another enzyme called HMG-CoA lyase. And what that simply is going to do is it's going to lop off a portion of this molecule, and we are going to make acetoacetate. So there's one of our first ketone bodies. And so the acetoacetate can be exported into the circulation, just like it is. But now if we go back to our graph and we look at the concentrations of ketone molecules, we see that acetoacetate is actually the smaller concentration of the two. Beta-hydroxybutyrate is the higher concentration. So acetoacetate is a rich energy-containing molecule, but we can perform one more reaction to make beta-hydroxybutyrate, and this is simply a redox reaction where we are going to take acetoacetate and further reduce it into beta-hydroxybutyrate using NADH. Now the reason we do this is that we're packing acetyl-CoAs into acetoacetate, so there's equivalent of 2 acetyl-CoAs in there, but there's also a functional group, it's a ketone structure on this molecule that we can reduce and add just a little bit more energy. So the whole purpose of this last step is just to fill this molecule with the maximum possible energy that the liver can do. This then gets placed into the circulation. Now once it's in the circulation, any cell can take this up, and any cell can use this for energy except the liver. The liver has no capacity to use these 2 ketones for energy. Now, what explains this drop in gluconeogenesis that is called the glucose sparing response? It's simply the decrease in acetyl-CoA because we're shifting now some of this acetyl-CoA into ketone synthesis. And so what we're doing is we're slightly decreasing the activation of pyruvate carboxylase. And so that means that we're pushing a little bit less substrate into gluconeogenesis, and that causes that decrease. And that's called glucose sparing. Now, why would you want to do that? Well, think about fasting out to this point, out to 2 weeks. We're going to talk tomorrow about the contributions of proteins and amino acids to gluconeogenesis. So the order of using fuel, we use glucose first, then we use fatty acids, then we start making ketones. But in order to make glucose, we have to get our substrates in. So if we start reducing our fat consumption, our fat level in our body, because we're using fat for oxidation, then we're going to start turning to amino acids. We're going to cannibalize ourselves. No other way to describe it. We're going to break down our proteins, and we're going to take all those amino acids, and we're going to start using them for gluconeogenesis. Is there a problem with cannibalizing your proteins too much? Yeah. Eventually, it's going to lead to death. Now, there's not really a big differential in metabolic substrates when you're going after protein. We go after protein in our skeletal muscle because we have more of it. Cardiac muscle it's different. Structure, function is different, but protein-wise, it's not different. It's protein. And so that can also be capitalized on. So the glucose sparing here is simply to start conserving and protecting our body from cannibalizing with these other substrates. And we're shifting to ketones. Now the problem with ketones is that one of them, acetoacetate, can spontaneously be converted into acetone. This reaction is simply a decarboxylation reaction, and no enzyme is required. So if you don't need an enzyme, that means that this reaction is going to follow the general rules of chemistry, and so the more acetoacetate you have, the more chemical conversion this is going to make to acetone. Hence why, early on a fast, if you're making ketones, you don't smell. But once you progress along the pathway where your ketone levels in your circulation start increasing, primarily acetoacetate, now you get to smell something. What happens in an uncontrolled diabetic? Fruity breath. Yeah, that's the acetone. So what's happening in this kind of biosynthetic cascade in an uncontrolled diabetic? Well, if you look at their glucose levels, here's what our response is. That's our normal response to glucose consumption. Theirs is like this. They start up here, they go up here, and they probably stay higher. So what this means is that they have an enormous amount of glucose in their blood. So they have an enormous amount of energy potential. The problem is that this is in their blood. And once it's in the blood, it's not useful. We cannot make energy out of this. So the problem is gotten around by these individuals by their bodies thinking that they are in a fasting state all the time. Even though they have high plasma glucose levels, they typically, in the beginning, before their beta cells burn out, they typically have high insulin levels. But if you can't get the glucose into your cells to use for ATP, your body's going to use the next source of energy, which is what? Before we get to ketones, fats. Yeah. So the diabetic is going to use fatty acid hydrolysis, beta oxidation, in the liver. They're going to think they're in a chronic fasting state. With increased beta oxidation, because they can't get to their glucose in the circulation, what's going to happen to ketogenesis? Yeah, it's going to dramatically increase ketogenesis. And it's for this reason here, is when an uncontrolled diabetic comes into a clinic and you smell that fruity smell of acetone, they are in a metabolic crisis at this point because they have so many ketones in their blood that what do you think their pH is going to be in their blood?
Low.
Very low. Okay? So that's what happens in an uncontrolled diabetic. All they're doing is they're experiencing a perfectly normal metabolic reaction. Their body is thinking they're in a fasting starvation state, and they're just using their alternative energy supply of fatty acids. Okay? Now, once cells take up this ketones, what do they do with them? Here's our primary ketone. The first thing the cells are going to do with this is convert it back to acetoacetate. And in doing so, the cell is going to capture that last bit of energy that the liver placed onto that ketone structure. Of course, this is all occurring in the mitochondria of the target cell. Once we have our acetoacetate, then we can convert this into acetoacetyl-CoA, and then we have a simple step reaction to make 2 acetyl-CoAs. And then these go straight into the TCA cycle for energy. Now again, any cell can do this. Do you think muscle cell will do this? Yeah, muscle cell is a goat. It'll eat anything. It'll gobble these. What's the most important organ that's going to use these?
The brain.
The brain, yeah. So this is all about the brain. And this is what's going to keep the brain conscious as those glucose levels start to decline in the circulation. Now here's the problem, again, with diabetic ketoacidosis. So for example, a 53-year-old male presents to the ER in distress. You measure their plasma glucose level, and it's through the roof compared to normal. Then you check the plasma ketones because you smell this fruity breath, the acetone breath, and sure enough, really high plasma ketone levels. So this person has metabolic acidosis, but it's ketoacidosis this time, not lactic acidosis. So here's our reaction that we're smelling the acetone. The acetone, of course, is volatile. It'll come out of the lungs very easily. And so what this person is going to do is rapid breathing. And simply the rapid breathing, Kussmaul breathing, is to try desperately to reverse that drop in pH in the blood. And this is the reaction that this breathing is trying to do. Here's your bicarb in the red blood cells. You're trying desperately to combine this with the proton to produce water and CO2. Exhale the CO2, but the most important thing is that you're trying to reduce the concentration of those protons. You're trying to increase the pH back to normal. So that's the effect of the Kussmaul breathing. Now, the other thing I want to remind you of is back from CVE pulm renal is the effect of pH on O2 binding. What's stimulating this rapid breathing response? Remember the Bohr effect. The Bohr effect is that decreased pH, so acidic pH, is going to increase oxygen release in tissues. So once oxygen leaves the red blood cell, leaves the bound hemoglobin for the tissue, CO2 primarily from our TCA cycle reactions is going to enter the red blood cell in this form. So what this reaction is going to do is it's going to assist by the increase in concentration of the proton. This is going to increase the oxygen release. So the more CO2 you have in the tissue, the greater the oxygen release because you're decreasing the affinity of oxygen for hemoglobin. And that's on this oxygen binding curve. So let's say normal pH here is pH 7 or pH 7.4, the middle line. Here's your normal oxygen tension curve. If we shift the curve to the right, because we've dropped pH, notice that shifting it to the right will decrease the affinity of oxygen for hemoglobin. And this is the Bohr effect, how it helps us release oxygen into our tissues. The problem with this is that it also reduces the oxygen affinity in the lungs. Because if this is our partial pressure of lungs, what we want is maximum affinity of our hemoglobin for oxygen in the lungs. And if that's up here, and we increase our pH in our blood, we are going to decrease the amount of oxygen bound. So our overall O2 sat is going to go down. Okay, so this is large part of the drop in pH, this ketoacidosis that's causing this increased breathing problem and oxygenation problem. So rapid breathing, trying to raise that pH to try to restore your normal oxygen binding affinity. Okay? Questions? Okay, so this is just a little bit of a hint of where we're going to go in endocrinology when we're talking about diabetes, getting into the physiology and then some of the biochemistry of diabetes and what the patient responses are and how you can treat it. Yes?
Does beta-hydroxybutyrate add anything to the acidosis seen in diabetic acidosis?
Yes, it's an acid.
It is itself?
Yes, both ketones are acids. So as you increase that, you're decreasing the pH, increasing the acidity.
Okay, so it's not mostly from acetate to acetone, it's because both ketones are themselves acids.
Exactly. The acetoacetate to acetone, that reaction is simply a byproduct reaction. It's pure chemistry because there's no enzyme mediating it, and it gives you a clinical tool to detect if somebody is in lactic or ketoacidotic stress.